mouth, esophagus, stomach, out the pyloric sphincter, and we are now into the small intestine. And when we look at the small intestine, it is three parts, three sections: the duodenum, the jejunum, and the ileum. The duodenum is the first part. It's only about 10 to 12 inches. Okay. It has some modifications to it because it is the first segment. Stuff that you see here that you don't see generally anywhere else. But the duodenum receives the acidic chyme from the stomach. It also receives some of the digestive secretions from the pancreas, from the liver, from the gallbladder. So the duodenum is the big kind of receiving segment of the small intestine. In the next segment, the jejunum. The jejunum is where the vast majority of digestion and absorption is occurring. Okay? A little bit happens within the duodenum, but really it's just more of we're secreting stuff in or it's receiving material. All right. The jejunum is kind of the big area when it comes to digestion and absorption. And it is all of this. Okay? And then the last portion is the ileum, the last segment of the small intestine, the last portion of the small intestine. There is some digestion that occurs here. There is some absorption that occurs here, all right? But for the most part, it all primarily occurs within the jejunum, okay? Jejunum, and then the ileum. Some of the adaptations that we see within the jejunum and the duodenum, we don't have here within the ileum. And we'll point out some of those here in a little bit. There is a little valve, a little sphincter that we find right here. And that is called the ileocecal sphincter. You can also see it written as ileocecal valve. It's the same thing. Okay, I will try to put sphincter on the exam since it's the one in the notes. But if you know it as ileocecal valve, okay, that's another name for it. This little sphincter or valve that you find basically right here is what governs the entrance into the large intestine or the content emptying out of the small intestine, however you want to visualize it. So here is, and you know, as you can kind of see, there's like 10 feet of material here all squished in your tiny little trunk right there. It's crazy. There is a ton of real estate here, but really when we look at the cross-sectional anatomy of these parts of the small intestine here, there are a lot of adaptations to increase even more surface area inside these tubes. We do have a couple of hormones that are secreted by the small intestine. As you can kind of see, they are written right here. And as you can see, the duodenum is the primary place for the major amount of secretion from these two things right here. Secretin and cholecystokinin, CCK. We'll see these a lot in the next unit as well. And we may talk about these again in the 4th unit as well. Secretin and cholecystokinin. Secretin does one major important thing. Secretin helps to cause the release of bicarbonate solution. Bicarbonate solution is going to come from a couple different places. One of the bigger places is the pancreas. Okay? Bicarbonate is a buffer. We have a very high acidic material coming from the stomach into the small intestine. We have a major need to buffer that material. The duodenum also has some other glands. So if you know what Brunner's glands are, yes, they're here. Okay. Brunner's glands you find within the duodenum. Brunner's glands also secrete a buffer material. But secretin is a hormone secreted by the duodenal cells, the cells that line the duodenum. And that triggers the liver and the pancreas predominantly to secrete a bicarbonate-rich fluid that helps to buffer that acidic chyme. So I don't have an ulcer here, okay? Cholecystokinin, bicarbonate, important. But cholecystokinin, come on, it's the big dude here. Cholecystokinin does a number of fantastic things. One, it helps to slow down the emptying of the stomach. Fantastic. Why is that fantastic? I don't want stuff coming out too fast. I want to be able to ensure that I fully, effectively break down this material, those macronutrients, into smaller and smaller and smaller bits. So by slowing it down, I can ensure that the small number amounts of enzymes that get produced by the liver, gallbladder, pancreas, when they enter into the duodenum here, that they can do what they're supposed to do. So I can absorb the nutrients effectively. Okay. So the slowing down the emptying of the stomach just ensures that I break down and absorb stuff properly. That's all that means. It stimulates the production of bile. The liver is who produces bile. And we'll talk about the accessory structures here in a little bit. But bile is produced by the liver, and this helps to stimulate the bile being produced by the liver. Bile is also stored in the gallbladder as well, so it stimulates the gallbladder to squeeze it out like toothpaste. Okay? It all comes down to toothpaste in the digestive tract. So it oozes it out, squirts it out, okay, because it's pulsable. So it squirts out a little bit of bile, helps us to break down fats. Cholecystokinin also stimulates that pancreas to create, or I guess secrete is probably the better way to say it. It's pancreatic juice. It's pancreatic enzymes. There are a number of enzymes that are going to be secreted by the pancreas. So we'll just kind of look at some of the major ones. So right now we have 3 hormones and a whole buttload of enzymes. Secretin, cholecystokinin, and gastrin. Those are hormones. Okay, those are hormones. They get released and they cause effect in a number of various other places. Enzymes are not hormones. Okay, enzymes are released by the cells to help break down stuff right there. So there is a difference between hormones and enzymes, which is kind of a hint. When we look at the small intestine, when we cut that thing in cross section and look at the fantastic anatomy here of it, there are a number of features, 3, that really help to allow the small intestine to do what it do. What does the small intestine do? Absorb nutrients. The digestion and absorption of nutrients is primarily occurring here in that dang small intestine. Okay? And these 3 features that I'm going to talk about here help to increase that surface area. So I can do more absorbing. And it increases, and I give the amount about 200 square meters. That doesn't mean a damn thing to you. But a tennis court, you can visualize that. That's what these 3 things do. They increase the surface area a ton, to the point that it's the size of a damn tennis court. It's about 600 times the amount of size that gets added to that one little tube. Okay, about a 600-fold increase in surface area by these 3 things. Holy smokes. Okay, so there's a lot of absorption. There's a lot of real estate here for the small intestine to do what they do, absorb stuff. Okay? Absorb stuff, primarily the jejunum. The first major adaptation that we see here that we really don't see anywhere else are circular folds. Okay, an old school name, if you've taken it, is plicae circularis. Okay. And all that name means is circular folds. These circular folds are there to, well, increase surface area, but really it helps to slow the movement of content through the tube. Yes, I have peristalsis, segmentation, all that happening here. But as this is going to, let's see if I have a nice, oh, I'll see if I can pull up some phys notes here in a little bit and show you a better picture here. I'm not a good drawer, so I apologize. So here's all these circular folds here. See, I stink at drawing. So here's all these folds. And as you can imagine, if you had to walk through a bunch of stuff falling down from the ground or from the sky here and coming up from the ground here, if you had to move through that, it'd take you a little bit more time to go through. That's what those circular folds do. Okay? Slows it down and allow us to absorb as much as we can. We have villi and then microvilli. Villi are tiny finger-like projections. That's all. Okay. So here on the circular folds here, if I did a circular fold, again, I'm not a drawer, right? You'll have a number of villi on top of those folds. Man, this is magical stuff right here. There you go. So there's all my little fingers. Okay. Again, I'm increasing the surface area for enzymes to be secreted. I'm increasing the surface area for absorption to occur. It's the only big reason. So this is your second major adaptation. The third one that we have here in the small intestine are microvilli, tiny hairs. Where do we see those? Again, oh my gosh, right? How awesome is this? This is the best picture in the world. I think my 8-year-old could draw better than this. Maybe. Yeah, he probably can. Okay. Here's microvilli. Again, surface area on top of surface area on top of surface area. Holy smokes. Okay. These little hair-like extensions come off of the intestinal cells, and that's what this is right here. So intestine. Okay, intestine cell. Okay, that's all enterocyte means. So I'm gonna use these terms interchangeably. So there you go, enterocyte or intestinal cell. All right. These are tiny little hairs that extend off the surface. Now, there are some better pictures of these two, and I'll show you those here in a second. But these guys do two major things. Again, more real estate for enzymes. And again, more real estate to absorb more stuff. All of these are to increase surface area, slow movement down, so I can do what I do here in the small intestine. Absorb, absorb, absorb. Let me go to this picture. So here's my circular fold. Okay. And here is my finger-like extension. Okay. And all of these little dudes right here, all of these little columns, this is simple columnar epithelium. Okay. Here is my intestinal cell. Okay. This is what lines all of these villi here. Oh, here it is right here. Okay. This is my intestinal cell or my enterocyte. And there's those tiny little hairs here. Okay. There are those tiny little hairs. These guys here will have what's called brush border enzymes, and I think I have that listed down over here. Yeah, brush border enzymes. Okay. This looks like a brush border. That's another way sometimes microvilli are referred to as in some textbooks, brush border. It looks like a little hairy, like top of your head. But here are my intestinal cells, my enterocytes. This book also calls them absorptive cells. Intestinal cells or enterocytes is how you'll see it probably on the exam. I know that's how you'll see it on the exam. But right there. And then here is our material. Here's our chyme. And again, all those enzymes that have been released from the liver, gallbladder, pancreas, come down here and act on this thing and break it down into smaller bits so I can have absorption to occur. As you can kind of see, epithelium, this is simple columnar epithelium. Right? This is epithelium right here. Right? This is stratified squamous epithelium. Okay? Many layers of flat shaped cells. But epithelium is what lines all the boundaries. This is the boundary. Okay, this is a boundary. Right? These are highly regenerative epithelial cells. Okay, highly regenerative. All right. As soon as I rub up against it, I'm sloughing off some cells. They are constantly dividing. There's a huge turnover here, and that's what I'm trying to show you here over here. They have a high turnover rate. So about every week, these are sloughing off. They're in your poop. Okay? So these are sloughing off. About every week, you're replacing all of these intestinal cells. So on all of these little villi right here, these things are being sloughed off very easily. And every 3 to 6 days, okay, about every week, you're having all of these intestinal cells renewed. A ton of wear and tear is happening down here. There's a lot of activity occurring here. We absorb all of these nutrients into this little intestinal cell right here. Once we get it inside this cell, I need it out. Okay. So it'll go through a mechanism of different things and get it into either a lacteal or capillary. So let's go back to here. So I absorb these nutrients from the out, pass them through my intestinal cell, and then on the bottom end, I get rid of them. I dump them in either a capillary or a lacteal. Okay, a capillary network is just a blood vessel. Okay, it's part of the circulatory system. It's a blood vessel. Okay, put blood vessel here. Okay, blood vessel. All right, some stuff goes into there. Some stuff goes into the lacteal. What goes into the lacteal? Lipids. Big lipids. Okay? Big lipids. What goes into the capillary network? Water-soluble stuff. Okay? Mechanisms upon mechanisms here, and we'll talk about that in the next unit. Okay? We'll talk about that later. But water-soluble nutrients and certain fats, small fats. Okay. Your bigger fats, they needed a little extra mechanisms to move them around, and so they enter the lacteal. Right. Whether it's going water-soluble or lipid, or capillary or the lacteal, ultimately it will get to the liver. Via the hepatic portal vein. Okay? And there it is. Okay? This is your hepatic portal vein. All, ALL, all of the nutrients need to go to the liver. The liver processes, packages, modifies nutrients, micro, macro nutrients, everything goes there. Stuff gets stored there. Right? The liver does a lot of stuff. Right? The liver does a lot of stuff. The ways in which I'm absorbing these nutrients into the intestinal cells can be many. Right? And if you've had 101, then all of this should look very, very, very familiar. Right? Simple diffusion, facilitated diffusion, active transport, osmosis, endocytosis. Sorry about that. These are the major ways in which we are absorbing stuff into these intestinal cells. Okay. The most basic method in which we are absorbing nutrients into the intestinal cells is via simple diffusion. Okay. And when it comes to simple diffusion, all it is just going down its concentration gradient, as you can kind of see over here. Excuse me, going from an area of high concentration to an area of low concentration. Right. This is the most basic, the most simplistic way that we are bringing materials into an intestinal cell. Okay. Most of your water-soluble vitamins are brought in this. I should put a comma right there. Most of your water-soluble vitamins are brought across in this method, where they just go from high concentration to low concentration in the luminal space inside the intestinal cell. Very high, obviously, there with the foods that you eat. Very low inside the intestinal cell. So this is going to be the method that those primarily go. Facilitated diffusion is just like simple diffusion, except it needs help. Okay. Again, we're going down the concentration gradient, down a concentration gradient, just like diffusion. Okay. So that's the same part. The part that's different, that it needs help. Okay. We need to facilitate. If we facilitate something, we help. Right. So here, this is going to be something that we have to go down the concentration gradient, it goes from high to low. But these are going to be bigger molecules. Okay, so they can't just easily diffuse across the plasma membrane without damaging it. So we have a protein carrier that facilitates that movement across the plasma membrane. That's what facilitated diffusion. For fructose, sugars found in fruits, they're going to be rich in fructose. This is the method in which they are brought across. Active transport is the exact opposite of the other two. Okay. Active transport goes against the concentration gradient. Okay. Against the concentration gradient. For these individuals, they need ATP. Okay. ATP is energy. Okay? This is energy. Okay. They need energy. They also need a carrier. So they're kind of like a facilitate, like we saw up there with facilitated diffusion. All right. To get across, to go from an area of low concentration to an area of high concentration, we go against that. It doesn't want to do that. Okay. It's not normal. But we got to move these things across. And we do see this quite a bit throughout the body. You do see this method of movement. Glucose works in this fashion. Okay. Amino acids work in this fashion. All right. Amino acids do a lot of things. They help make structures. We're constantly breaking down and recycling amino acids to make things. So inside of a cell, there's always going to be a lot of them. So we need to have active transport mechanisms to bring those across. The last one we have is osmosis. Osmosis is a special case of diffusion. It involves water. Okay. It involves water absorption. So it's going down, okay, high concentration to low concentration. A lot of water over here, less water over here. So water is going across. So when we bring water across, which again, water is a component that, we need to, ensure that we get enough of, and we have to absorb in. We can do that through a number of different areas. It's not just in the small intestine there. So, but it does happen predominantly there. Osmosis is the movement of water. So for all of these, osmosis is the one that is unique to really just water. Endocytosis this is a mechanism that's on here, but it's not really common in terms of what's happening within the small intestine for absorption to occur. This is when the material gets close to the cell, and then we engulf it. We bring it in that way. Okay, it's like cell eating, as you can kind of see there from the picture there at the bottom. This is how proteins. This is how infants absorb proteins in milk. But once we stop consuming breast milk in the first 6 months or a year or whatever, then endocytosis is not a huge thing that we need to worry about. So endocytosis, again, maybe an example of these may be on the exam. Definitely a brief explanation of it. It would be as a multiple choice type question on the exam is how you would see these things. But these are the 1, 2, 3, 4 major ways, 5 different ways in which nutrient absorption methods occur.